Good morning. I'd like to welcome you to the Electrical Engineering Colloquium. This morning, we're very pleased to have our brand new Assistant Professor, Sreeram Kannan, to talk to us about applications of information theory to a variety of topics. Sreeram comes to us from his postdoc at Berkeley, and before then, he was at the University of Illinois, Urbana-Champaign, where he got his PhD. He's had some distinguished experience in award-winning research, including the Van Valkenburg Prize, as well as the Qualcomm Cognitive Radio Contest. Sreeram, thank you so much for speaking to us today.
Thank you very much, Professor Chansar. Good morning, everybody. Thanks very much for coming to my talk. As introduced, I'm Sreeram, and I'm a new assistant professor here. So I view this more of an intro to all of you so that you can stop by my office to talk about whatever you want to. So let me put this work in the context of what electrical engineering is in my view. If you think about the last 50 years, we have witnessed many tremendous contributions by the electrical engineers. And here are a few of them. The integrated circuit. Here is the internet, satellite networks, and wireless communication. All these have fundamentally revolutionized the way we interact and the way we deal with machines. And if you think about what is common in all of these, it becomes very clear that the scale of complexity that we deal with in order to make a single chip work, billions of transistors interacting, many, many millions of routers working together in the internet, many satellites, and similarly many towers working together. So in some sense, electrical engineering can be thought of as complexity engineering. How do we engineer systems with high complexity that can interact with each other and produce results that are useful to mankind? So if you think about it, one may be tempted to think maybe we are the masters of complexity engineering. However, a single look at the cell will dispel this illusion. So the cell is indeed if you look at biology, the amount of complexity that goes into making the organism work is tremendous. Even at the scale of a single cell, you have so many genes interacting in order to make the cell work. So in this context, it's very natural to ask, since electrical engineering is complexity engineering, what can we say about biological systems? And I'm going to pose two questions. The first one is, can we apply these electrical engineering principles in engineering biological systems? Like, can we create an organism which does something that we want? So this first question forms the field of synthetic biology, and we have professors in our department who are studying this. The question I'm going to focus on mainly is the second one, which is, can we use the principles developed in our field of electrical engineering to reverse engineer biological systems? Which means you have something happening in a biological system, and by making observations, can we try to find out what is going on inside? So in this context, one may ask, what are the observations we have in order to make these reverse engineering assessments? And one can see that our ability to look inside the cell over the last decade has multiplied manifold. And this began in 2001 with the completion of the human genome, where we sequenced one human genome basically at the cost of $1 billion. And today, in 2014, the cost of sequencing a single genome has dropped several orders of magnitude to $1,000 per genome. So here, there is a comparison shown between the cost drop in genome sequencing compared to Moore's law, which is well known to be the driver of electrical engineering. And you see that this is not even exponential growth. This is super exponential growth. So this ability to look inside the cell at this resolution has given us a tremendous ability to acquire information. But the question is, how do we go from just bare information into something that we can say is knowledge about the biological system? So this is the question that drives the one portion of my research, which I'm going to talk about today. So I said we have acquired a tremendous ability to sequence the DNA and RNA in cells. However, there is a simple caveat here. The thing is, if you have a long DNA sequence, our ability to sequence many, many reads is very high. However, whenever we get an observation, we cannot sequence the whole DNA as it is, but rather we can only get fragmented views of the DNA. So you take the DNA and fragment it into small pieces. Like, you take a paper and shred it into small pieces. And from these pieces, you basically want to reconstruct the original DNA. So this is called shotgun sequencing because you're just taking the DNA and then applying this shotgun method to divide it into small pieces and sequence this. So the data that we get from our sequencer is of the form of these short views, or short reads, as they are called in this field. So in some sense, if you really wanted to know what the DNA is, you have to do two things. One is first take the DNA, and then you subject it to this shotgun sequencing method, which will give you short reads. But then, in some sense, you need to have some software which will take these short reads and produce you the long DNA sequence back. So in some sense, we can view the sequencer itself as having two components. One is a hardware component, which basically goes from which basically gives you observations of these short reads. And then there is a software interpolation component, which will take these short reads and produce the long DNA sequence. So in some sense, the construction of this sequencer is a clear engineering problem, not a biological problem. And this is where I think is one of the places where electrical engineers can play a major role in the development of both the sequencing hardware and the accompanying software. In our group, the main focus is on algorithms and mathematical methods that drive this reconstruction software. How do you go from these short views in order to reconstruct the long DNA sequence? So this forms the main thrust in our work. So I said that we have acquired a tremendous ability to sequence the DNA. However, this has had many unintended consequences. Unintended originally, but turns out to be blessings in disguise. So for example, we can now not only sequence DNA, but also many, many molecular mechanisms. Like, for example, you want to know what is happening in the cell right now. You can sequence the RNA. The RNA gives you a snapshot, like the memory of a computer, like the RAM of a computer. It will tell you what the cell is doing right now. You can also, using the same methods of DNA sequencing, basically use this as a primitive in doing many other tasks. Like, for example, you want to find where proteins will bind to the DNA and start transcription and other processes. Or you want to know how the 3D structure of DNA is, whether several genes are close together in three-dimensional space or farther away, and many other molecular processes. And all of these can be done inside this same framework of DNA sequencing, followed by the reconstruction software. And what really changes in all of these different methods is the way you prepare the sample, which goes before the hardware, and finally, the way you construct your reconstruction software. And like I said earlier, our main work in this area is in the development of this reconstruction software. So how do we approach this problem? So our work falls into three subcomponents. And the first component is coming from this information theoretic lens. So we can ask basic questions, for example, of when do we have enough information to go from these short reads to get the original DNA sequence? This is something very basic we can ask. Because, for example, if you think that all these reads are too short, like, for example, the most trivial case is all these reads are of length 1, then clearly we don't have any information to reconstruct the whole sequence. I forgot to mention that when you get a small subsequence, you have no idea where this subsequence is coming from this long sequence. So in some sense, it's all jumbled up. So the first question we may ask is, what are the informational limits? Which means, when do you have enough information to reconstruct the sequence? The next one is, can we develop efficient algorithms to solve this problem? And finally, we need to have a software package that can basically operate with the output from the sequencer and create the desired results. So this is our general this is a general overview of the kind of work we do in our group. And now I'm going to focus, so I have two parts in the talk. Depending on the time, I will see how much I can cover. The first part is about RNA assembly or RNA reconstruction. This is joint work with Leo Pachter and David Shea at Berkeley and Stanford. Okay, so like I mentioned, so in this project, the main aim is to know what the RNA is in a given cell. So let me just quickly review the basic dogma of molecular biology, which is that the DNA, which is you can think of as like the hard disk, contains all the information necessary for the running of this computer. However, the RNA is the one, whenever you need to do something with the DNA, you first convert it typically into RNA. And this RNA gets converted into protein. And this protein is the functional work cause of the cell. So in some sense, if you look at a particular cell or different cells in a given organism, they all have the same DNA sequence. However, they all have different RNA because they are doing different things at the same time. For example, the gene for eye color will not be expressed in the skin cell. So if you sequence the skin cell, you can start finding out what genes are getting expressed in the skin. So this has many important applications. There are clinical purposes. For example, you can take a healthy cell and a diseased cell and see what the differences are between the two. And this can tell you potentially what is going wrong in which gene. And it's also useful in research, for example, in the discovery of novel functions in many different applications. This is one example of a salamander which has very good regeneration abilities. And if you want to know why it's able to regenerate its limbs, for example, you can sequence the portion which is regenerating and try to see what the RNA is there. And many other tasks like understanding gene regulation. Okay, so what is the, let me go to a more precise definition of the problem. In a given cell, you may have many different RNA. So this is RNA1. So they are all basically so each RNA is some subsequence of the DNA cut and pasted in a certain way. So here is one particular RNA, and here is another one. And each of them may be present at different concentrations. So it may be now different number of copies in the cell. So, and when you sequence this, what do you get? So you can think of the sequencing process as doing the following. You're going and picking out a random RNA from the cell, maybe this one. And once you pick this out, what you're doing is essentially getting a subsequence from that portion. So here you sequence this one, and you get the sequence ATT. And similarly, the next time you pick out a random one, and you get this. So after the sequencing process, all you have is this bunch of reads, which are basically subsequences from these different sequences. And the goal of our reconstruction software is to take these small reads or small views without any knowledge of where each read came from which RNA or which position in the RNA. And from this, how do we reconstruct the list of all these sequences and their relative proportions? And here, there are many existing software that can do these tasks. However, then one may ask, why do we even study this if there are existing things that can do it? However, if you look at how well they agree, so here is an example where we take the same input, the same set of reads, and feed it to different software, these three software, for example. And we ask, if we look at the list of output RNA and then see how many, for example, the intersection here, 9741, basically is the number of RNA that all these three assemblers agree on. This one is predicting 400,000 RNA, which are not predicted by this, not predicted by this. So you can see that the three different assemblers are not even agreeing on the total amount of RNA. Forget the actual list of them. So here we raise an important point. The question is, are these assemblers disagreeing because there is an informational bottleneck, which means your observations are too small or too short for you to be useful in actually reconstructing? So is this an informational bottleneck? Or maybe there is no informational bottleneck, but there is a computational bottleneck, which means there is enough information, but these software are not able to exploit the information within the given time frame of computation to solve this problem. Or maybe it's neither. So Our main results, like I explained earlier, our viewpoint is essentially to first find the informational limits. And as I illustrated in the previous slide, this is very important because you want to know whether you have enough information to actually be able to reconstruct the data. And we can do this. We can establish the informational limits. And we also come up with efficient algorithms that approach these limits. And finally, we also have a practical implementation. So I will go over this in somewhat more detail later on. But basically, ideas from information theory, graph theory, network flows, and sparse signal recovery work together in order to create these solutions. So let me just illustrate what I mean by an informational limit. So here is the space of all read lengths, right? So like I said, your observations are basically short pieces of your original RNA. So we call it reads. And the read length basically says how long these reads are. And we assume here that, let's say you have many, many reads, since that is not the bottleneck. However, you're constrained by your read length, which means what the size of your reads are. Then we can ask, is there a critical phenomenon, which means, is there a certain point called L critical such that if your read length is smaller than this, then you're information limited, whereas if your read length is larger than this, then you have sufficient information to reconstruct. And in fact, we can show that this dichotomy holds, not in a very general case, but in almost all instances of practical interest. So we can, for example, characterize that this threshold for read length, for example, in reconstructing, let's say, mouse RNA from mouse is actually something like 4077. We have also similar calculations for the human transcriptome, where the critical read length is 10,000. So one question immediately that comes up is, this critical read length, is it met in practice? Which means, do you get observations of length greater than 4,000 in practice or smaller than 4,000? It turns out that typical read lengths hover around 100 to 200, not 4,000 to 10,000. So the question remains, what can we do at practical read lengths? So we flip the question on its head, and what we ask is, we say you are given a fixed read length, and ask, let's say we cannot reconstruct all the RNA. Maybe it's okay. But what fraction of these RNA, they're called transcripts, what fraction of these transcripts can we reconstruct? And we can show that the information bound and the algorithmic performance are quite close. And most of the existing algorithms which do not exploit a fact called abundance diversity actually need to have a performance lower than this. For example, at read length 100, you see that there is a tremendous amount of difference between the existing algorithm's performance and what we can achieve using the new algorithms. Okay, now I will go to the third part, which is the software. And so we have built a software package encouraged by these theoretical results. And this software package is capable of assembling a large amount of reads. For example, in this experiment, actually, it says 20 million reads here, but the latest experiment I have is 135 million reads. And what we see here is, so here what we have plotted is the fraction of transcripts reconstructed. And what we see here is across different quantifications, we see that our algorithm, which is on the rightmost, performs much better than all existing algorithms. And this holds true across a variety of different settings. Okay, so let me go back to let me give you some idea for how this work is done. So like I said, you have a list of RNA, and these RNA all have different proportions, and we are going to call these proportions, they're called relative abundances, but these proportions are alpha 1, alpha 2, and so on. Now, what we have is observations which give you short reads, and these short reads you can also potentially estimate. For example, this read, this sequence doesn't appear anywhere else in the list of RNA, and therefore this read appears at proportion alpha m, whereas other reads, for example, this portion, this blue portion is common between the two RNA S1 and S2, and therefore if you sample this particular read, you're likely to sample it at proportion alpha 1 plus alpha 2, because either you sample transcript 1 or transcript 2, you get the same sequence. Okay, so this is the model, and the question is, when can you reconstruct the list of sequences from these list of reads? So let me demonstrate how we compute the informational bottleneck. So let's take these two transcripts, the first, which basically share this portion S3 between them. There is no overlap in the first and the last portions. So if you have these two transcripts in your system and you observe reads, I've placed the reads on top of these transcripts just for illustration. Okay, so now here I've just replicated this, these two transcripts. However, let's say I swap something there. I swap S5 and S4 in this right transcript system. Now, the claim is these two transcripts, these two systems of transcripts can produce exactly the same set of reads. Why is that? Any read that is produced here from this portion, you can as well assume that maybe that read was produced from this portion, because this read also aligns to this portion. Similarly, any read that comes from the middle portion, this S3, can clearly be assigned here. Any read that comes from S2 can be assigned here, and so on. So as long as your reads are shorter than the portion S3, which means there is no read going from S2 to S3 to S5, now you cannot disambiguate between these two possibilities. So in some sense, given short reads of this form, there is no way for us to distinguish whether this is the transcript system or this is the transcript system. And so unless the length of S3 is smaller than your read length, these two transcriptomes can be confused. So this highlights that your length and your repeat structure in your transcripts basically interact in such a way to create certain confusions. Okay, so now the question remains as to how we can basically distinguish between these two hypotheses. So like I mentioned earlier, different transcripts in reality actually appear at different proportions. In this example, I have shown them to be occurring at exactly the same proportion. So let me correct that, and in fact, let's make sure that different transcripts appear at different proportions. Maybe the first transcript is rarer than the second transcript. Now let us see if the confusion still holds. So here is the alternate, so here are the two hypotheses. Here is the original hypothesis, which is that these are the two transcripts. And here is an alternate hypothesis that these are the two transcripts. The only difference between the two is that S5 and S4 are swapped. Again, actually, any read that comes from this system can actually be mapped to this system. However, if you look at this explanation for your reads, as opposed to this explanation, this one looks more satisfactory, because here the reads are appearing in uniform proportion throughout the transcript, and whereas here, the reads are staggered in non-uniform proportions. So in some sense, the fact that different transcripts have different abundances can be used in order to disambiguate between the two hypotheses. You can say that this is the right answer, not this one, because of the fact that this displays a uniformity which this doesn't. So this is a very important signature, and this is what we call as abundance diversity, taking view from the same kind of diversity that was useful in a wireless channel. Different people have different channel strengths, and this can be used to transmit to different people to the highest one first, and then you can transmit to the next highest, and so on. So the same kind of idea can be useful basically in disambiguating between the list of transcripts. Okay, so let us see how this algorithm basically leverages this kind of information, which is abundance diversity, in order to solve the problem. So so the only information we have is the list of reads, right? So here is a list of reads that maybe the algorithm gets. And from this list of reads, the algorithm tries to find patterns of overlap between them, because that's the only signature we have in order to reconstruct the original sequence. So we construct something called a transcript graph. And how does this work? So we take the list of reads and find overlap patterns. For example, let's look here. So these two reads share this pattern ATTC, ATTC. So one may hypothesize that maybe these two reads came from adjacent portions of some RNA, and therefore draw an edge, right? So what I've done is I take the reads, and then I find overlap patterns between the reads, and then I draw edges between the reads based on the overlap patterns. Now you go and look at some other two reads, and you find some overlap pattern, and you make these connections. So in this way, one can get a graph, right? You take the list of reads, and from this list of reads, we create a graph, which we call as the transcript graph. And we can also do some more things. For example, we can ask, how many times did I see this overlapping portion in the set of reads? And then you can weight these edges based on these relative frequencies. So in essence, what we have done is we have taken the reads, and from that created a graph, which is the set of interconnectivity between the reads and the weight of these interconnectivity. So this is what we call as a transcript graph. And we can simplify this graph in some ways, which I will not get into. OK, so now the question is, from this transcript graph, how do we get the list of RNA or the list of transcripts? So the way we do it is, one simple way is that you basically try to enumerate the list of paths through this graph. And here there are two paths, one going upwards and the other going downwards. And these two you can maybe decode as the list of paths. OK, so now let's see if this is all you need to do. So let's take this example. Let's take this example here. And in this example, you see this graph. And now there are 4 possible paths in this graph. The first path basically goes like this. The second one goes like this. The third one goes like this. The fourth one goes like this. So there are 4 possible paths through this graph. But the question remains, do I need to enumerate, do I need to look at all these paths in order to explain this graph? OK, so one may say that you want to use the minimal number of paths that explains this graph. So one possibility is to basically say that these two are the paths. However, the other possibility is to say that S1, S5, and S2, S3, S4, these are the 2 paths. How do I disambiguate between the two? The important signature here is that the relative weight here is the same as the relative weight there, and the relative weight here and here are same. So you can use this to basically say that these are the 2 transcripts and their relative abundances. So here is a basic idea, which is I use the weights in order to connect together the various paths. So this is an interesting first idea that we can start with. However, if you go to somewhat more complex graphs, this idea does not work anymore. So let's see. So in this graph, you see that the weights on these nodes. So here you see 12, 9, and 6, and here 5, 7, and 15. So if you try to match up the weights, then you may match up 12 to 15, 9 to 7, and 6 to 5, because they appear close enough. However, if you look at these numbers more carefully, you can find out that 12 is actually exactly 5 plus 7, and 9 plus 6 is exactly 15. So in some sense, the minimal hypothesis which explains this graph is to say that these paths, the red paths, which comprise of these transcripts, basically explains all the weights in the graph, because now this transcript plus this transcript explains the upper portion of the graph, and these 2 transcripts explains the lower portion of the graph. So in general, what we have is the following problem. We have a general graph, and for this graph, we have weights on the edges. And given this graph and weights on the edges, we want to find out what the paths are through this graph that explain these weights correctly. So in this case, we can see that the numbers here all are 0.12. So you can say maybe there is a path here of weight 0.12 and a path in the lower side of weight 0.88. And these two basically explains this graph fully. So in general, what we want to ask is, given a set of edge weights like this, what is the fewest set of paths that can explain these edge weights? Right? So this problem occurs in various contexts in network routing, snooping, and so on, where basically the question is, for example, in network routing, you may ask, what is the fewest number of paths do I need to use in order to send information optimally from one source to the destination? And it also occurs in other contexts. And OK, so since it occurs in other contexts, people have studied this before. And they've shown that this problem is actually NP hard. So the NP hardness basically means that in the worst case, there are some hard instances. So it means that these hard instances, unless P equals NP, may take actually super polynomial time. So if that is the case, then even if you have enough information, your computational complexity for finding these fewest number of paths is going to limit you from solving the problem. OK, so what we did is went into this hardness and figured out that the hard instances actually occur when most of the paths have exactly the same flow value. So if most of the paths have exactly the same flow value, that is equivalent to saying that almost all the RNA have exactly the same abundance, which means they're all present in the same proportion. However, this is not at all characteristic of the biological problem. In the biological problem, different RNA have typically different weights. And how do we resolve this problem? So what we do is we assume that the abundances are generic. What that means is that with very high probability, two of the abundances, for example, are never equal. So if we assume such a condition, then we can obtain a provably correct algorithm that reconstructs the list of paths under a condition on the read length. And under this condition, the algorithm runs in linear time. So this is quite an interesting fact that actually, even though the problem is NP hard, by using some biological assumptions, we are able to find an algorithm that operates in linear time, which is quite stark. And the way the algorithm runs is essentially by using the fact that the abundances are generic. So instead of going into the details of the algorithm, I'll just show you one example run of the algorithm. So here is a graph, and this graph has various weights on these edges. And since I've written it out in a certain way, it's obvious that the path A goes like this, the path B goes like this, and the path C goes like this. And that explains this graph. However, the algorithm is not presented with the numbers like this. It doesn't know, for example, this number is A plus B, and so on. So what the algorithm does is it tries to decompose paths going through node by node. So let's say I go to node 3 and look at the local graph structure around node 3. Then you see that the numbers are basically A, B plus C, A plus B, and C. And it tries to ask, what are the fewest paths locally that explains these numbers? And it turns out you need 3 paths locally to explain these numbers, but it's not unique. It's not the only one that you expect, that A goes like this, B goes like this, and C goes like this. There is another one also. So the algorithm says, I don't know what to do, so I'll go to the next node. So it goes to the next node, and node 4 turns out to be somewhat of an easy node. You have A plus B coming in, A going out, B going out. The only way to explain this data is to say that A goes through the upper path and B goes through the lower path. So what we have done here is to condense the graph in a certain way by unraveling the paths locally. So we know now 4 and 6 occur together at frequency A, and 4 and 7 occur together at frequency B. Now, once we have done this unraveling, the algorithm proceeds and tries to go back to the original node, which it could not resolve. Node 3 it could not resolve earlier, but now if you look at node 3 has the following structure. It has A and B plus C coming in, and A, B, C going out. And if you ask, what are the fewest number of paths that explains this? It's only 3 paths, because A has to go like this, because it's clearly matched, and B and C have to be split between the upper and lower paths. So essentially, you get this graph, which says 1 goes to 3, 4, 6 at frequency A, and so on. So from this, we basically obtain a list of transcripts or list of sequence of nodes and their relative weights that corresponds to this graph. And we can show that the algorithm which proceeds in this way actually obtains the right answer. And to go from this simple theoretical setup to the practical algorithm involves many, many steps. It nearly took me 1 year to build from this initial theoretical results to the actual software. And there are many, many steps that are involved in this. I will just highlight a few of these. One of them is basically the model that I presented till now assumes that the reads have no errors. However, whenever you get these partial views, they're also partly erroneous. So we have to do some interesting algorithms for error correction. The second one is, how do you estimate the weights on this graph? So the weights on these graphs are estimated based on the reads and their relative frequencies. However, they should also satisfy the property that numbers add up in a certain way to become a flow. So we have to use some algorithms, in particular some convex min-cost network flow problem in order to solve this maximum likelihood problem. And the other one that I did not cover is whenever I said the algorithm proceeds locally by trying to ask what are the fewest number of paths locally that explains this problem. And it turns out that we have to use some randomized L1 relaxation algorithms from sparse flow theory to solve this. And finally, we also need to solve a feedback arc set problem to deal with cycles in the graph. So this is just to give you some idea of the tools that are needed to make this algorithm work. So I'm going to skip this, and so I will conclude the RNA part by saying that we had an initial theoretical problem, initial practical problem, which was basically that you had all these different software which did not agree with each other in practice. And we modeled that as an informational limitation problem and calculated the information theoretic limits for that problem. And then showed that there is potential information in the problem which is not exploited by these other algorithms, and constructed a new linear time algorithm that achieves this, and finally built a software that is capable of exploiting this information. So the next part of this talk, I'm going to focus on DNA reconstruction. And this is joint work with Sohail Mohajer, who's a faculty now in Minnesota, and David Shea. OK, so like I pointed out earlier, DNA sequencing has exploded. Our ability to sequence DNA has exploded. But also, our interest in sequencing DNA has exploded. There are many, many species that we want to sequence. There are all the individuals. It'll be nice to know what the DNA differences are between all the various individuals. And even though typically it's assumed that a particular organism contains the same DNA in all the cells, there are some variations called somatic mutations, which is as the cell differentiates from the original zygote to the various cells in the body, there may be some errors. And these are quite critical in understanding various practical problems, medical problems, like, for example, cancer is mainly due to mutations in the DNA. So as our ability to sequence genomes have exploded, so has our interest in sequencing a variety of genomes. So as you observe, however, that all the different genomes we want to sequence are all not independent. There is some correlation between the different sequences that we can potentially exploit. For example, if you want to sequence one species of a fruit fly, having sequenced another species of a fruit fly should give you a lot of information. Similarly, having sequenced one particular person's DNA should be much easier to sequence some other person's DNA. OK, so this is the problem we focus on, which is called reference-assisted assembly. So what is it? So you have the target which you want to sequence. And like earlier, you obtain short reads from this target. And these reads are fed to the assembler along with a side information of maybe a related DNA, maybe from some other person in the same species or some reasonably related species. Or maybe if you want to sequence a particular cell in the body, you have a reference as some other cell in the body. So this assembler basically gets this side information, and we want to know what the utility of this side information is. Can we now start doing much better if you have a reference than if you didn't have a reference? So an immediate thing that we need to know is how is the reference and the target related? Because if the target can be arbitrary relative to the reference, then there is no information clearly in knowing the reference. So we need to catalog what the types of variants are.
So one type of variant is called a SNP, which is basically substitutions. So you can see the blue portion here is replaced by some red SNP. So the A is replaced by G and so on. Almost all the other bases are the same. Another type of variation is insertion and deletion. So you see that the reference had, from the reference, there is an insertion to get this portion, insertion to get this portion, a deletion to get this portion, and so on. This is another type of variation that's common in humans. There is also more severe variations, which are called structural variants. You take the reference, and maybe the two portions are swapped, or maybe one portion is duplicated, or maybe it's duplicated in different portions, or maybe just the same thing is duplicated many number of times. So all of these form different types of variations. And in prior work, basically, there are two problems. One is that algorithms are tailored to a certain type of variant, and the other is there is no agreement on when an algorithm works, and there is many debates on these things, and there is absolutely no theory to explain this problem. Okay, so information theory has been very successful in dealing with different types of side information. Like, for example, if you want to compress something, what is the utility of knowing something else? So these questions are quite common. And so we want to ask, what is the utility of this side information? And what are optimal algorithms that can help you utilize this information in the best way? So here is the algorithm. And what does the algorithm get? The algorithm gets a reference DNA, which is the DNA of a related species. And maybe it gets some measure of relatedness between the original reference and the target. So for simplicity, we are going to focus on the case of SNPs, which basically means substitutions. And we also get many reads. So dense basically means we are in the regime where there are many, many reads, and we are constrained especially by the read length. And from this, we want to estimate the target DNA. So let me rerun between what the relationship between the reference and target is. So here is the reference, and here is the target. And as you can see, these triangles basically show that in these portions, the target differs from the reference. In this case, in the simplest way, by a SNP. This particular nucleotide may be T instead of an A in the reference, and so on. So now, how many such variations can be there between the reference and target? For this, we define this moving average distance, which is basically saying take a window of length m, and in this window of length m, there are no more than k differences in any given window. So this window, as it slides through the reference and target, you see that there are at most 2 differences in any given window in this example. In general, you take a window of certain size, and in this window, wherever you keep the window, there are at most k differences between the source and target. Okay, so in this context, we want to ask, like I said, what are informational limitations in this problem? So let's take a reference, and like we saw earlier in the RNA problem, what confounds you in this problem is repetition, repeated sequences that appear in the sequence. So for example, here you see that this yellow portion is repeated in this reference DNA. So now let's consider 2 possible target sequences. So the first target sequence looks like this. There is a SNP in the middle, a green SNP, and here there is a red SNP in the middle. So I will, now if you look at it, the reads, this particular SNP gives rise to a certain set of reads which contain information about the green SNP, and these reads contain information about the red SNP. Now I'm going to make an assumption that your read length, the read length is smaller than half of this repeated region size. The L repeat is bigger than twice the read length, which means there is no read which gives you straddling information between this green SNP and the local region. And similarly for the red SNP. So now what I can do is I can go and create, so like I said earlier, there is no read which gives you a connecting information between this green SNP and the local region. Okay, so now I can create an alternative hypothesis T2 where the 2 SNPs are swapped. The 2 substitutions are swapped. So this hypothesis also explains equally well the set of reads, because these green reads can be explained based on this region, and the red reads can be explained equally well based on this region, which means if you have reads from this or this sequence, they are identical, and there is no way for you to distinguish whether the original true sequence was T1 or T2. So this is an example of an information limitation when your read length is smaller than the repeated region over 2. Then the two targets are indistinguishable from the reads. So we can do some more accurate analysis, which basically says that even if these two portions are not exactly the same, but are approximately the same, this problem continues to hold. Okay, so what could be an algorithm that uses the reference DNA in order to assemble the reads? So let's take this reference DNA, and let's say the reference DNA has this structure. So there is a region, and there is another repeat, basically just like the earlier example. Now there is a target DNA, which is the one you want to sequence, and this has some SNPs relative to the original reference. So let's say the SNPs are occurring in certain portions, and you get reads from this system. And now let us assume that your read length is bigger than half the repeated region size, because if it is less than that, we know that no algorithm can reconstruct the sequence. Okay, so now you have this, and you have reads from this sequence. Now what a simple algorithm that one may think of is to take the reference DNA, which looks like this, and try to take each of the reads and see where they could map approximately into the reference. So let's take these reads, and since this region is not repeated anywhere, these reads map into this particular portion. So if you take reads from here, however, they could map potentially to this portion or to this portion, right? Because these reads could have come from this portion or this portion within the distance prescribed. However, there are some reads which have straddling information which could have only come from this portion. And similarly, this is uniquely mapped, whereas this is multiply mapped. And some reads are uniquely mapped. So if you try to use all these reads to try to call the basis, then at this portion, we may call this green polygon. In this portion, we will call this red diamond, maroon diamond, and this, and so on. However, that is not correct. As we can see in the target, the diamond doesn't appear in this portion. So one thing we can do is to keep only uniquely mapping reads, that is reads which you're sure where they belong. Right? There are some reads which belong only to this portion, and using that, you can estimate the DNA sequence. So it turns out, under this condition, you can actually estimate the DNA sequence. And this is a very simple algorithm that basically succeeds under the theoretical condition that we proposed earlier. Okay, so this algorithm is somewhat unsatisfactory because it's doing the most trivial thing, which is taking the reads and seeing where they could have come from this reference DNA. And in some sense, in some theoretical sense, we can show this algorithm is optimal. However, it's not quite satisfactory because in practice, there is something that you can do better. And actually, we can repair the model to bring this into the theory. So let's see what can be done. So let's take this example. Here is the target DNA sequence, and here is the reference. And let us say now you failed the condition that we proposed earlier. Then what happens is you take the reads from this target, and they map. And if you only keep uniquely mapping reads, there are no reads. So here, all the reads map multiply. They map either here or there, and these reads map either here or there. So since these reads map multiply, there are no uniquely mapping reads in the middle, and therefore there is no information for me to decode this under the old algorithm. So the question that arises is, do we have to throw away all the non-unique reads, or do they give some information that can be used in an intelligent manner? And it turns out almost all existing algorithms only work with uniquely mapping reads. So here we are asking the important question for practice, which is it possible to improve these algorithms? Okay, so let's go back to this example and see that in this region, there are no uniquely mapping reads. What can we do? So like we know from here, there are some multiply mapping reads in this region. Can we use them in some particular way, right? So let's go here, and let's bring back the reads a few at a time. So let's bring back reads that only mapped in this portion. So these also mapped multiply, but I don't care about it, and I bring all of them back. So let's say this read and this read are brought back here. Now if you look at these two reads, we can ask which of the, so using this uniquely mapping read, I can decode till this portion of the sequence, right? So I know exactly what all these bases are. Because if a read uniquely maps to a certain portion, I can decode it without any error. So now if I decoded this portion correctly, now, and these two are the possible reads that can sit on top of this, now I can decide whether this is the correct read or this is the correct read. It turns out that I can eliminate this read because this read is having a SNP on this portion, whereas I know that there is no SNP in this uniquely mapping read. So I have a pivot, which is a uniquely mapping read, and I've decoded this portion. And using this pivot, I can decode other reads. So I can say that these two reads, among these two reads, this is the correct read. And now, once I know that this is the correct read, I can decode the sequence all the way till this green point. Now I bring back the reads, the other non-uniquely mapping reads, and there are these two possible reads. This read says throughout it is yellow, whereas this read says there is green at this portion. I know that this is the correct read because I have already decoded this green portion. Now I can bootstrap my decoding like this and keep proceeding till I actually decode the whole of the sequence. So this is a new algorithm which basically performs what I call as iterative alignment. So you first align and use the uniquely mapping reads, and from this you've decoded a certain portion of the DNA, and you can use this to start decoding other portions. This kind of an iterative algorithm is quite common, for example, in decoding LDPC codes and so on. So it turns out that even this algorithm is not optimal, and we have a new algorithm which we call as iterative multibridging, which has come out of the theory, which basically does two different things simultaneously. The first thing is to use the reference DNA in order to try to solve as much of the problem as you can. And then the second part is to forget the presence of the reference and solve the de novo reconstruction problem, which is where you don't have any side information. What is the optimal thing to do? You do that, and then you iterate between the two. And it turns out this algorithm can actually do much better than the earlier algorithms. And there are actually, we can show somewhat matching necessary and sufficient conditions, which don't always match, but our conjecture is that the proposed algorithm is optimal for real genomes. And we're working on this currently. And to summarize the DNA variant calling part, we proposed two mathematical models for studying this variant calling. And we showed that we proposed a new algorithm, iterative algorithm for variant calling for the particular substitution error model, but it also generalizes to other kinds of variants like insertions, deletions, and structural variants, which offers a unified framework. So I'll conclude this portion of the talk here, and just in the next minute or so, point out to future directions. So here I've laid out a typical structure of how a wireless network is structured. So there is a physical layer, network layer, and application layer, and also other layers in between, which I have not pointed out here. But the basic goal of the physical layer is to bring data input from the electromagnetics and decode the signal. And at this output, you have a cleaned up digital signal, like, for example, bits that come out of the physical layer. And the network layer deals with network level organization and so on. And the application layer deals with how to use the network in order to provide a useful practical application. I'm going to draw an analogy between this and architecture in computational biology. To me, sequencing is like the physical layer of computational biology. Because what you get at the sequencing is from the physical hardware, you get some information with partial views and noise. And you have to decode this information in order to get the, let's say, the DNA sequence or RNA sequence and so on. And once you get the DNA RNA sequence from this data, what you want to do is some kind of a regulatory network inference. How do these genes interact? How do the various portions in this biology work together in order to create a coherent system? And we can ask basic questions similar to these in this context, which is how much information is needed to do reliable regulatory network inference? What are optimal algorithms to do such inference? These are directions we are working on in our group. And finally, some applications of this to solve real problems where, for example, you mine not only the regulatory network and the sequence information to be useful in understanding, let's say, disease pathways, aging, and so on. And finally, I would like to conclude by saying that electrical engineering and algorithms from information theory, signal processing, and graph theory have a big role to play in reverse engineering these biological problems. And I'd like to conclude my talk with that. Thank you.
Thank you very much, Dr. Kannan. We have time for one quick question. Yes, right here.
So the approach you described to reconstruct the genomic data, like from the transcripts that you got from the sequences, could similar approaches be used to, say, reconstruct the sequence in a way that would allow for more manageable storage and analysis?
Oh, so that's a very good question. In fact, that's one of the, repeat the question. So the question is essentially about whether the same techniques can be used to not only reconstruct the DNA and RNA sequences, but also to maybe compress and do other operators on the sequences. It's in fact a very important question, and it turns out that many of the current consortia are actually suffering from a huge data deluge. And like I described, for example, the reference-based DNA assembly, you can also do reference-based DNA compression. Like, for example, I know my DNA, and let's say I get reads from John's DNA, then I could store them in relation to my DNA, thereby getting a huge amount of compression. So these are techniques already people have started using, but what the basic information theoretic limits are for these problems, people don't know. So those are, again, very interesting directions. There is also another direction where, so sometimes it's okay that you have enough space to store your data. However, what is lacking is you don't have enough memory to load your data. So there are very interesting questions in what is being popularly called compressive genomics, which is, can you somehow do algorithms also in this compressed space so that so even though I've stored data in a very compressed form, I have to decompress it, load it into my RAM, and then do whatever operations, for example, comparing DNA sequences and so on. Can I instead just load the compressed data into memory and work in the compressed space to obtain interesting results? And there is already some beginning work, but again, the basic questions remain completely open.
I think that we'll thank our speaker here. If you have further questions, please come down after the talk. Thank you very much, Sreeram.
Thank you very much.